is a nightmare.
Activate.
Okay, so we're gonna start looking at analyzing the questions for the biology paper for 2018, and that'll be paper 1. We're looking at the higher tier for triple. Okay, going into there, we're gonna have a look at the answers for the questions, but we're also going to try to figure out why those answers are correct. Okay, so eating food containing salmonella bacteria can cause illness. 2 symptoms of infection by salmonella are vomiting and diarrhea. What causes these symptoms? Right, so your answer for that will be either toxins, poison, okay, from the bacteria. Question 1.2. Give 2 ways a person with a mild infection of salmonella can help prevent the spread of the bacteria to other people. So for that, we can just write something simple as wash hands before cooking. We could also write down isolate yourself. Okay, a couple other things that we could also mention would be disinfect the clothing and surfaces. Do not share utensils with other members of the family or with other people. You could also write down wash your hands after using toilets. Now, moving to question 1.3, it says, in very serious infections of salmonella, a doctor can prescribe drugs to kill the bacteria. What type of drug can a doctor prescribe to kill the bacteria? And your answer for that will be antibiotics, okay? Now remember that antibiotics only work on bacteria, they do not work on viruses, and that's why they're not prescribed for when you have a cold, unless you actually have a chest infection, which actually it will be bacterial as well, and then you can get prescribed the antibiotics. You could just write names of some antibiotics like penicillin, amoxicillin, or anything else like that, but you just better safe by saying antibiotics, okay? For question 1.4, a person with AIDS may take longer than a healthy person to recover from salmonella infection. Explain why. Now, the reason for that is because the immune system is damaged or is compromised. Okay, that's going to give you 1 mark. And the other mark would be that the white blood cells cannot kill that bacteria that creates the salmonella, or the salmonella bacteria. Okay, this may be by various reasons. It could be because they don't have enough white blood cells because the immune system is compromised, or because the immune system is not working properly, and that's why the white blood cells cannot get rid of all of that bacteria, so it just takes a little bit longer. Question 1.5. Salmonella bacteria can be transmitted from chickens to humans. Chickens can be vaccinated to prevent the transmission of salmonella bacteria to humans. Suggest 1 other way farmers could prevent the transmission of salmonella from chickens to humans. Okay, so some of the answers could be to give the chickens antibiotics. Another answer could be to just don't sell infected chickens or eggs. And also, it could isolate the chickens. Okay, so note that this is a 1 mark question. Any of those answers could give you a mark, so you have quite a few different possibilities in order to get those marks. Okay, now a restaurant owner employed a scientist to test the effectiveness of 2 chicken cleaning liquids. The scientist took samples from 2 work surfaces before the surfaces have been cleaned with the cleaning liquids, after the surfaces have been cleaned with the cleaning liquids. The samples were analyzed for the numbers of bacteria they contain, and the results were shown in figure number 1. So this is what we have. On the y-axis, you have your number of bacteria, and that is your dependent variable, because that's what you're counting. And your independent variables are the things that you change. So you have cleaning liquid A in the sink and table, and you got cleaning liquid B in the sink and table as well. That's before cleaning. That's the dark one, before cleaning. So you can see the number of bacteria in the sink and the number of bacteria in the table are pretty high compared to after they were clean with cleaning liquid A. And so in the other table as well, in the cleaning liquid B, you can also see a high number of bacteria in the sink and a high number of bacteria in the table before being clean, and then after being clean you have these numbers over here. Okay, now clearly we can see that if this is going all the way to 50 and you got 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 boxes, that means that every single one of those tiny little boxes is 5. Okay, is equivalent to 5. Okay, so that way means that right here in the middle we would have 25. Right, that's just a point of reference for us to realize that. So over here we got a 25, and over here we're gonna have 45. Oh, 20. Sorry, that would be 20, and then 20, and then we have 20, 20, 25, 30, 35, 40. I think that would be 40 over here, and this one is going from 50 to 55, and that's halfway through the dot, so that would be 57 and a half, maybe 57, 58, but number of bacteria times 10 to the power of 10. So let's have a look at the question. Question 1.6. Which cleaning liquid is more effective? Give the reason for your answer. So obviously in there, cleaning liquid B will be the best option. Cleaning liquid B. And why is because you can see a greater reduction in the number of bacteria in both locations. Okay, that will be that. For this, you have to have both of them correct in order to get that mark. Okay. The scientists investigated the effect of cleaning liquid A and cleaning liquid B on the Salmonella bacteria grown in a laboratory. Okay, so we have figure number 2, and this will be a Petri dish. You have a filter paper dish with soaked in water, filter paper dish soaked in cleaning liquid A, filter paper disc soaked in cleaning liquid B. A Petri dish was placed in an incubator at 25 degrees Celsius for 48 hours. After the 48 hours, the scientists calculated the area around each of the paper discs where no bacteria were growing. So then you got results are in table here. So an area around the disc with bacteria growth in centimeter cubes. So around this disc, there was no area in which the bacteria didn't actually grow. So that's why you got a 0 in there. Okay. There you go. So you have a 0 in there because no bacteria, there was no area around this here, around the water, where bacteria didn't grow. Okay, so it was all full of bacteria. Then you have cleaning liquid A, which is this one here. Okay, and it says area around the disc of 11 centimeter cubes. So if we can just maybe just do that for the purpose of the example, that would be 11 centimeter cubes. Within that area there, no bacteria actually grew. Okay, around that red area there was no bacteria actually growing because the liquid the cleaning liquid A was in that piece of paper, so it stopped the bacteria from growing. Now when we look at cleaning liquid B, that was in this one here, it says that it has an area of 13 centimeters square. So just for the purpose of illustrating that, obviously 13 is bigger than 11, and again this is not to scale, so you will look at something like that. Okay, so there was a bigger area around the cleaning liquid B, or the paper that was soaked in cleaning liquid B, where bacteria did not grow. Okay, so what measurements would the scientists need to take to calculate the area where no bacteria were growing? And this is a 1-mark question, and for this you will be looking at the radius or the diameter around the paper. Okay, so there you go. Right, question 1.8. Give one change to the investigation that would allow the scientists to check if the results are repeatable. Now, the only thing that you can actually do, and the question itself is telling you the results are repeatable, will be to repeat the same experiment again and just look for similarities. And then we got question 1.9. The scientists showed the results to the restaurant owner. Both cleaning liquids cost the same per decimeter cube. So just one other factor the restaurant owner should consider when choosing which cleaning liquid to use. Well, it's pretty easy. You will look at the toxicity levels of those cleaners. Would they be affecting the health of customers? Would they be affecting the health of the employees in the restaurant? How long does the cleaner actually remains the area keeps the area clean? How the efficacy time is? Interaction with other cleaners as well, how they react with other things that you use in the restaurant or the chemicals. So some of these things are the things that you could actually look into before investing in buying in these detergents. Okay, so for your answer, all right, so to continue now, we're gonna go to part 2 for question number 2. So click on the next video so we can continue analyzing the answers for the higher paper, Triple Biology 2018.
It was a nightmare. Activate.